Today we are going to talk about genomic research and Ayurveda. So, genomics is a very wide branch. What specifically genomics is trying to do in medicine is there are two, three things that genomics does in medicine. First is personalized healthcare. Personalized healthcare, uh, it is something like so when you start giving medicines to someone, whatever anyone has, even if it is the same disease, diabetes, for example, if we take same two individuals have same diabetes but they'll not respond equally. They won't have same diabetes, same type of diabetes also. So many things will be different because between two individuals in normally also general sense, two individuals are very different. Eye color, from the eye color, hair color, body build, everything is different. Even the choice of food and what they like in terms of weather, anything. If you say there is a lot of differences between two individuals. So genomics, if you see this here, there is this DNA strand. This is made of proteins called ATGC. A is you might have heard of all these four things ATGC. So these proteins, so wherever this bond is made between two proteins, A and T or G or C. So wherever there is a difference between two individuals in any of these areas in a gene, then it is called a variation between two individuals. So there are around 10 to the power 23. 10 to the power 23 means there are 23 zeros after 10. These many differences are there between two individuals. So there is a lot of variability, and when you see between individuals, neither they are similar baseline. Normally, when you see if they are healthy, they are not similar. And when you check uh, in disease state also, since they are not similar in baseline, when disease comes also it affects differently to every person. That's why there is the need of personalized healthcare, because same medicine doesn't work similarly on everyone. And then it comes disease prediction. Two people can have same kind of food, same genetic predisposition also, but still they will get that disease differently. Two people living in the same house, same city, doing everything same, one can get disease early, other can get later, one can not get disease at all. So, disease prediction, who will get what disease? And then pharmacogenomics means pharmacogenomics is made of two words. Pharma means drugs, whatever we give them, any kind of treatment, and genomics means genes. So there are specific genes identified which are linked to specific drugs. Like there are many drugs. If you have studied modern medicine, there is warfarin that is used to thin blood. Warfarin is associated with a type of SNP variation in which that person responds differently to warfarin than the other person who does not have that uh, variation. So, pharmacogenomics is the third thing. Now, traditional medicine, when we say traditional medicine, how it can um be merged with genomics, there are a lot of things to be done in traditional medicine, especially Ayurveda, because Ayurveda, first thing you know, it is personalized healthcare. We know that we always do Rogi Pariksha first, then Rog Pariksha, and we see that whatever is suited for this individual, then that only we give for the medicine. We don't give everything to everyone, same thing to everyone. We'll tailor the needs of that individual and then give that medicine only to that individual according to the desh, kal, prakriti, the Dashwit Pariksha we are doing, everything we are doing, and then only we are giving medicine. So personalized healthcare is ingrained in Ayurveda and other traditional medicine systems also. Now, how it is linked. So what we do is we do experiments and see that how it is linked with genomics. Can we find any uh, similarities, or can we find things that we can map to genomics or any other molecular science for that matter, not just genomics? And then there is the holistic approach. Like if we integrate genomics. We have found signatures. We'll talk about these signatures later. But if we have found something that is related to genomics and Ayurveda, then we can merge both of them. We can see how both of them we can use together to treat any disease or anything whatever we are treating. So Ayurgenomics, as Prerak said, this is a new branch of science. Mostly, you'll call it a transdisciplinary science because it is a blend of Ayurveda and genomics. And we are our lab in IJIB. We are working mostly on this and the Trisutra axis, the whole Trisutra axis, Hetu Ling Aushad, and we have the lab started from Prakriti route. Right now we are also doing in experiments and studies in diseases and not only in healthy individuals in Prakriti but also in diseases. Globally also there are a lot of things happening, especially in traditional Chinese medicine. You can see a lot of studies that have been linked to traditional medicine practices. Genomic studies in India have been done since 20 years now. And in traditional Chinese medicine also, you can find many kind of genomic studies, plant-based genomic studies, non-plant-based, human-based genomic studies. So there is a lot of things going on. Then there comes genomic research in Ayurveda. What is the potential? Potential is molecular signatures. First thing is whatever is written in Ayurveda, like Prakriti, Dosh, Dhatu, Mal, all those things are there. But how do we explain them in terms which other people understand? Now, you and I both understand English. But what if somebody comes, what if I don't understand Nepali at all and you are talking to me in Nepali and I'm not getting anything, not even a single word what you're talking about, then I cannot get what you are saying and or nor can I respond to you. I cannot understand what is the value of what you are speaking. So if we are talking in terms of dosh, dhatu, mal, if any scientific person who has no idea what these things are will never be able to understand. So what we do is we take small things from Ayurveda and we research about them and try to find molecular signatures, like what is related to these functions or these biological processes in our body, these types of gene processes in our body, these type of these things in our body. And we say that okay, these kind of characters when they come together it is a what feature. Then when we say this, somebody will be able to better understand us rather than just saying that okay, this is what. So this is what it's meant by finding molecular signatures from Ayurveda. Then there is personalized treatments. Now those molecular signatures or whatever findings that we have found, now applying those into treatment. First, we have found that what are the differences between individuals, how we can map those differences to Vatpit curve or Prakriti or other entities in our body, and now using all the findings into the treatment. The third thing is pharmacogenomics. Pharmacogenomics, not only the drugs that are there in modern medicine, but also the drugs, because in Ayurveda also herbal components are made up of chemical constituents. There are chemical entities which are naturally found in every plant. Those can have different effect in different individuals. If we can map those ingredients of the Ayurvedic medicines also to specific genotypes, specific genomic signatures, then we can see that how some kind of drugs affect only some individuals and other kind of drugs affect only other individuals. And this can be used in pharmacogenomics of Ayurvedic formulations also. Then comes precision medicine. Precision medicine, what is precision medicine? When you are applying all these findings of pharmacogenomics, of personalized healthcare, and precising the treatment for every individual, what that particular individual needs. Genomics as well as Ayurveda together, then it is called precision medicine. Precision means correct, exactly what is needed. So that is precision medicine. Now, I'll start what we are doing in Ayurveda. What has been done in our genomics since a long time, since 20-25 years now. Examples I'll give you of researches. So, what is the translational framework of Trisutra? Trisutra, you know Trisutra, Hetu, Ling, and Aushad. Hetu, Lakshana, Aushad. Now, when you connect Hetu and Lakshan, you see that Hetu means whatever is there that is causing something. Causes can be seen in basic biology, and from basic biology, what we are seeing in clinical settings are when the patient comes with some kind of symptoms or whatever it is suffering from, we can see that. So that is a clinical setting. So from clinical to basic biology, from from basic biology to pharmacology, Aushad. This is one axis, and the third axis is clinical medicine, combining the disease and the Aushad part, and that is the drug part. Now, this whole thing can be applied in clinical applications also for predicting for predicting what an individual can suffer from. So, preventing that thing, whatever we have predicted, that if somebody is predisposed or vulnerable to having somebody is vulnerable to having obesity or diabetes, then preventing that through diet, lifestyle, then personal, if in any case that prevention does not happen and that person gets any kind of disease, then personalized treatment also, and in preventive also there will be personalized care, because that individual needs specific exercise also. Every individual does not need same exercise, same kind of sleep, same kind of food, same amount of water. So prevention also will be personalized. Participatory means every individual will participate in their own health needs. And promotive health. Promotive means you are promoting health through Ayurveda and precision medicine. Then systems biological understanding. What in research, where all we can apply this is systems biological understanding. Systems biological understanding, it means that systems biology is when we are not taking every organ or every small thing as different, we are taking it as whole and see that how environment is interacting with our system, how our system is interacting with itself, the organs together, like Ayurveda does. It does not put everything aside. Even if it has given Mansik Rog different and Hriday Rog different, but when you see any disease, there will be description of effect on heart, there will be description of effect on the mental status, there will be description, even in mental diseases, there will be description of effect on other body organs and other systems of the body. So it is a holistic medicine that we say always. Then we can use it for innovations, like any kind of um, new kind of therapies. There are a lot of therapies that are there in Ayurveda, but there is difficulty in doing it. Specifically panchkarma therapies, you have so much difficulty in doing those therapies, so you can use in innovations to make new kinds of instruments, new kinds of things to uh, implement into innovative science. They for complementary or modern medicine. Now, novel discoveries. You can find some new things. I'll show you one example where we found a new thing in our research. You can find biomarkers. You can find new drugs, novel therapies, any kind of these things. So all these are used in translational research. Then we come to what we are doing for Prakriti. What we do is first we um, assess the prakriti of an individual. Prakriti assessment, you all know what is prakriti, how it is different, seven types of prakriti, what all are there. And how we do prakriti is every prakriti is given their prakriti lakshanas. What has its own specific lakshan, Pitta has its own specific lakshan. So according to that, we have made a questionnaire. We have prepared a proper questionnaire. We use that questionnaire and we do prakriti parikshan. According to that, we assign that Prakriti to individuals, and we only take those individuals who have a dominant type of Prakriti, either Vata dominant, either Pitta dominant, or Kapha dominant. Dominant means somebody who has more than 60 to 65% of one particular dosha Prakriti. Then we take those individuals and we do there are so many different experiments that we do. We also do structure and function activity. Like we use machines that are there is body composition analyzer, there is respiratory machines like spirometry, there is normal blood apart from normal blood pressure, we have different types of other machines also that can measure our blood flow, vascular thickness, and how good the blood perfusion is to some organ or some part of our body. Then there are skin parameters like skin thickness, skin luster, skin hydration status, like how much water content is there present in your skin, erythema content, like redness in your skin, melanin content, the chemical that gives the color of the skin. Then there are other things like body composition analyzer is one thing which measures fat mass, muscle mass, bone mass. There are a lot of instruments that we measure things physiologically and physically. So we do the structure, function, and activity part of it. Then what we do is we do hematological and biochemical parameters, all the blood tests from normal blood tests which are related to blood parameters, CBC, these all the blood sugar, liver profile, kidney profile, all those things. And apart from those, apart from the hematological parameters, biochemical and other enzyme parameters also, even the insulin levels, the homocysteine levels, all those parameters, blood tests we do. Then what we do is we check disease biomarkers through genomics. Genomics mein we what we do is we take the blood sample, we isolate the DNA, we isolate the RNA, we isolate the protein, and from DNA we do genomics. We see genotype, we do whole genome sequencing. The whole genome is sequenced, and we see what are the differences between two individuals. Or what we do is genotyping. There are specific kits available where we can only see what are the known genotypes available in the literature, and we check for those. If this is present in one individual, if this variation is present in another individual, these are the genotypes that we check. Then there is transcriptome. Transcriptome is called, transcriptome means, so when DNA is transcripted into RNA in our body and messenger RNA. That messenger RNA, what it does is goes and gives instructions for various functions that are happening in our body. Even if though two individuals have similar kind of genotype, that transcription when it happens, that can be different. Some person will generate with the same amount of DNA also, one person will generate more protein, the transcription will be more in one person, the other, the transcription might be less. So gene expression is what we check from transcriptome. Then there is metabolome. Metabolome means uh, all the chemicals in the body that are there, small chemicals, large chemicals, we test for those chemicals. It can be biochemicals, it can be hematological, enzymes, proteins, all the chemicals that can be measured are checked in metabolome. Then microbiome. Microbiome is human body is not made of just human body. We have multiple microorganisms in our gut, in our lungs, in our skin. There is skin microbiome, there is genital microbiome, there is gut microbiome, there is oral microbiome. So a combination of bacteria, they live in our body and help us by various ways. They interact with our body, they secrete some things, they use up some things from our body, they help in discarding the waste. So many things those microorganisms do in our body. So it is like a symbiotic relationship. So that the what is the composition of that specific microbiome also affects our body. So every individual has different microbiome. So we can study that microbiome also. Then there is epigenome. Epigenome is anything which affects the DNA. DNA itself is there. One person has a fixed DNA, but throughout life, some things are there that are affecting some places in that DNA, which affects the transcription of that DNA into RNA. That is epigenetic, these are called epigenetic changes. The sequence will remain same, ATGC, whatever is there, it will remain same, but the binding of that DNA, so many things are involved in this working of DNA. In that, one thing is that since a cell is very small, DNA, if you open it up, it will round up the whole earth. It will become such a long strip. So it is packed into compact chromosomes and fitted into that cell. Cell is very small, microscopic. So in every cell, the whole DNA is fitted. So that fitting also affects a lot. How the DNA is being transcripted into an RNA and how it is translated into proteins and how it is conducting all the functions in our body. All those things are very, it is a very complicated process. So those packings and packing proteins and all those things are also affected by things in our environment, things that we take. So all these things that affect that is called that packing thing is called epigenome, and whatever affects it, it is called epigenetics. Now there is the physiome. Physiome is whatever the physiological functions of the body is, and combine all the when you study the physiological functions of the body, like heart rate, autonomic nervous system, your cardiovascular system, these physiological functions when you are studying, all of them put together make up our physiome. So all these studies are conducted differently and then they are mapped together and seen the correlation between them. And what is these correlations have to do with Ayurveda or Prakriti. All these things in Prakriti individuals, these are the things that are done at an individual level. But other than that, at molecular level also, we do a lot of things. We take specific tissues and work on those tissues that are tissue specific responses. That is what we see. Then we take individual cell lines. So what we do is we take blood cells. From those cells, we take some specific cells and grow those cells in petri dishes in lab. So when we are growing those cells, we keep them in a conducive environment which is equal to whatever inside the human body is 37 degrees Celsius temperature, oxygen, the perfect oxygen what is needed inside the body for those particular cells. All things are controlled and those cells are grown in lab. And now we can do experiments on those cells and see if different types of Prakriti individuals, if we are taking those cells from different types of Prakriti individuals, will those at cellular level also they'll show differences when we do experiments on them. Now I'll talk about some studies that we have already done and what we found. So first study that was done was genome-wide expression and biochemical correlate. Genome-wide expression means I told you that DNA is transcripted into RNA, and that is called gene expression. Now, what we see from gene expression is there are different types of expressions for different pathways. When we do whole genome expression, all the genes in our genome, how they are expressing themselves, irrespective of what was their original uh, sequence, is seen in expression. So what we found was these are the differences that we found between these three uh, types of individuals, what, with, and cough. This study was done in healthy individuals. So what we see was in biochemical means all the blood tests that we did, we saw that in healthy range also, what individuals had high prolactin levels and prothrombin time. Cough individuals had high lipid profile. It was not high, it was not unhealthy high. It was within the normal range, but still it was significantly higher than Vata and Pitta. Higher lipid levels, higher serum uric acid levels, low HDL levels, and low prothrombin time. In Pitta individuals, what we found was higher HB levels, hemoglobin, PCV packed cell volume, and RBC count, red blood cell count. Now, the second thing is whole genome expression. When we do gene expression, we get a list of genes which are expressing. So if a gene was expressing there, so when we do experiments, the gene expression measures the content of RNA. If the content of RNA was in some microliters. Then it measures before in an individual. It sees that this was the expression in one individual, this was the expression in another individual, and we see that this expression is different from this how many fold. If this is one, this is two, then we see that this one two is twofold, yani twice the difference is one. It is double in the second person. So we see, we take all those genes together and we see what are those genes working on. So we take out the processes. These are called biological processes, like cell cycle. Cell cycle means whatever the the cell growth cycle. Then there is transport. Some genes which are involved in transport. Transport means transport of oxygen, transport of nutrients amongst the cells. These are biological cellular processes. Then olfactory transduction. Olfactory transduction is when the signal of smell transfers from nose to brain. This was found to be higher in Vath individuals. Regulation of enzyme activity. Whatever enzymes are there working in our body, their regulation is higher in Vath individuals than any other two. Low immune response. Whatever genes are working in immunity response, they were working lower than what was found in Pitta and Kapha. High immune response was found in Pitta. Lower olfactory transduction, that is the signal of smell that transfers from nose to brain, that was low in Pitta individuals. Cellular biosynthesis. Cellular biosynthesis means the um, the biochemical components that a cell produces to use. It can be glycogen cycle, glucose to glycogen conversion, or and the production of ATP, ADP, all those things. Cellular biosynthesis, macromolecular metabolism, that is at the molecular level, the metabolism, like when we metabolize fats into fatty acids, complex carbohydrates into glucose, all those things. And low fibrinolysis. Fibrinolysis means when the um, fibrin protein is activated in the blood, it is used for clotting, for mending. So this, it needs to be dissolved. Otherwise, what happens is fibrin reduces the collagen and all those things in different organs, and when the fibrins start depositing everywhere, it will become scar tissue. It will not be a healthy tissue. So once fibrins activity is done, it should start lysing. It should get removed from that place, and healthier tissue should start growing after any injury happens, whether be it on skin or the internal organs also. So it should lyse. But in cough individuals, it was found that the fibrinolysis activity is lower. So from these things, what we can see that what the individuals, if they have a low immune response, they might be prone to getting sick very frequently. If Vata and Pitta individuals both are subjected to the same kind of virus, if the virus is going on, influenza virus or normal cold and cough is going on in the environment, then Vata individuals will be more vulnerable to that than Pitta individuals, because their immune response is higher, their immune response is lower. Similarly, olfactory transduction, if we see, then what individuals will be better at judging things by the smell perception. That is, they can be really good um, in terms of, there are a lot of career options also nowadays. No, so you can say that they can be very good judges of smell perception. So they can go into perfume industries or food industries, that people and because of that transduction, their memory, olfactory memory, will also be stronger than other people. Then there is low fibrinolysis. Low fibrinolysis means the development of scar tissue, the vulnerability towards development of scar tissue. When scar tissue develops, what happens? Pulmonary diseases like pulmonary fibrosis, diseases like liver cirrhosis, these kind of diseases happen. So in case an injury happens, in tough individuals, that person will be more prone to not healing properly or getting a scar tissue more than the other two individuals. Then I talked about discovery, how you can discover new things. So this is another study that we did. In this study, what we found was there was a difference between cough and with individuals in one genotype. Genotype means at one point in the whole genome there was this uh, in EGLN1 gene. This is the gene which is hypoxia sensing gene. Oxygen. When we get oxygen into our body senses, right? If we are getting less oxygen, our body will sense it and it send the signal to the brain, and the brain will tell that you are getting less oxygen. Then we'll have breathlessness. These are the responses that our body does. So for so this is the very important gene which senses oxygen in our body. So when we checked the genotype of puff and in pitta individuals, there was a specific genotype which was first thing EGLN1 gene. That gene itself had expression more in cough and low in pitta. Then when we found that what gene that gene does, it was seen that that gene is responsible for oxygen sensing. This was not available first in the literature. So since we saw that there is difference in between cough and pitta individuals, what we did is we tried checking what that gene does and what is the difference between these two individuals. And then we got to know that EGLN1 is involved in high altitude adaptation, because what happens is we are people from high altitude. You are from Nepal and I am from Uttarakhand. So we are from higher altitude in the Himalaya region. People who are native from Delhi or any other place that is here down in India, these places are low-lying places. Low-lying places have higher oxygen than the and lower pressure than the what is found in our region. So we are adapted to that low levels of oxygen and higher levels of pressure, but these people are not adapted to that. Now, when these people go to those places for any work or uh, anything, for excursion or entertainment, for anything, if they go there, how well are they going to adapt to those changes is what this gene tells. Now, what we saw is the cuff individuals, when they traveled from here to there, they were they could not adapt very well, with the individuals could adapt much better. Now, when the genotype of with the individuals was matched with the genotype of people that are already adapted there, that are the natives of that place. This study was done in Leh, Ladakh. So from there also we took people, recruited people and checked whether their genotype is similar to any of these. So it was similar to fifth people. These fifth people who have never been there, they have no relation to that region, but still they had a genotype which was similar to them. So this made us discover a new uh, function of this gene. Earlier, it was only known as the oxygen sensor gene, but we found a new discovery was made that this gene is involved in high altitude adaptation. That is, this particular genotype in this gene is involved in high altitude adaptation, which helps some people to adapt really well there, and other people which do not have this will go there and get sick faster. They are more likely to develop high altitude pulmonary edema kind of diseases more when we go there. So this was about discovery. Then we can come to personalized drug response, pharmacogenomics I told you about. We have done studies in which, so there are different types of genomic studies. There are parts of genome. Okay, there is a whole genome which is called when we sequence that, it is called whole genome. Then we take only the differences that are known, that is called genotyping. Then the third thing is exon. Exon means only the whole genome does not code for protein. only specific parts, only 1% of our DNA codes for protein. Other 99% works in different other things. So when only that 1% is called the exon of our DNA. So of our out of our genome. That exon is those genes in that exon are specifically responsible for coding for protein. So they are directly involved in that coding for protein. So when we need to check only that, we do exome sequencing. Only that portions of the genome we take out, and we are testing only that. So when we did the exome sequencing, we found that there are 28 SNPs in 11 FDA approved genes. There are already genes databases that have vast information on genomics. We are linking this to Ayurveda. So what we saw is that whatever is there available in genomic literature, out of that, in 28 SNPs, we found pharmacogenomic relevance. CYP2C19, I was telling about warfarin. This is the gene that is responsible for response to warfarin. So basic genotype differences we found EGLN1, and there is one I told you about EGLN1, but there is one more VWF, von Willebrand factor. It is a thrombosis factor in. You have seen there are 12 factors of coagulation in our blood. So there is this VWF, von Willebrand factor, that is responsible associated with thrombosis. That is responsible for clotting our blood in time. What will happen if our blood doesn't clot in time? We'll start will bleed to death. Anyone will bleed to death. So this is very important. But what happens if it misfunctions? If it misfunctions, what will happen is it will start clotting things inside the body also while the blood is running through. Then what will happen? Thrombosis will happen. It will block the vessels of our body. So this is one important gene where we also found that it is different between cough and pitta individuals. In pitta individuals, there is a risk of bleeding, and in kapha individuals, there is a risk of thrombosis, which is connected to EGLN1 because high altitude pulmonary edema usually is developed with relation to thrombotic effects. So when cough people go to high altitudes, the maladaptation is caused by both of these genes. These are two connected. So this was another finding that we found. Now, these basic genotype differences will contribute to the differential disease susceptibility. We know the disease susceptibility, they are susceptible to pulmonary edema, with individuals are susceptible to bleeding disorders. So now we know that some kind of individuals are susceptible to some kind of diseases. Now we also know that some kind of genes are responsible for getting different kind of response from drugs. So one individual is, if we take the example of VWF, with the individual who is susceptible to bleeding disorders and also has this genotype which is responsible to warfarin, this individual is already susceptible to bleeding disorder and has a pharmacogenomic marker which is highly responsive to warfarin. So warfarin are blood thinners. They are given in case of some thrombotic effects. So if that person is highly responsive to warfarin, and in some case that person is given that drug, that person will react more. Already it has a bleeding tendency, and it has more reaction to that drug. So if we know all this beforehand, if we know the prakriti, we know these two differences, then we can adjust the therapeutics accordingly. This is what we mean by using whatever findings we are getting everywhere into what into therapeutics, into clinical settings. This is the example that I wanted to share. Then what apart from this, what we have also done is we have collected a lot of data of Prakriti. Prakriti data, what we do is now we have machine learning algorithms. We can now we don't have to sit and see what is the Prakriti of an individual. We have questionnaire. We enter that data, whatever is there from individuals. We take that data, we put into the machine. The machine will tell what is the Prakriti of that individual. We don't need a person to check the Prakriti of an individual. We can use the machine to do it. How we did it is, whatever data we had collected first, we took that data, we take every feature in that data and we mark it with this is what feature, this is pith feature, this is cuff feature, and according to this, the Prakriti of this individual is what, or this Prakriti is pith, this Prakriti is cuff, this Prakriti is what pith, this is pith cuff, this is, we label all that data and we feed that machine data into the machine. That is called the training. What we do is then we run, there are different models that we run. So not only in biology, but in machine learning and technology also, there is a lot of things that we can do. This is what I meant with innovation. So we feed that data into the machine runs some algorithms, it builds algorithms, it takes out different sets of it. So Google when we how is Google engine built? When we put a picture of a cat, Google will tell you which cat it is. How was it built? To build it, what they did is, in the beginning, they took hundreds of pictures of cats and told the machine that this is cat. This is cat, 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 cat. Then it took hundreds of pictures of dog and told that this is dog, 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 dog. And the machine started identifying it. This is what we do when we teach a kid. Small kid, when we give it a new thing, we'll tell this is this. This is a cat. This is a dog. This is an apple. Everything we teach that kid will start identifying. This is what machine also does. Machine will start identifying things and it will make its own rules, which are called algorithms, to identify things. And then what we do, when we have trained it, we'll give it testing set. 10% of that data we'll give it to test. And it will test and keep on testing and keep on testing and then tell us this is the result. If there is anything wrong, we'll tell it. We'll take out prediction accuracy every time and we'll say this is the prediction accuracy and we need to change these models accordingly. This is a long process. I'm just giving you an example that this is how innovations are done, not only in biological settings, but in technological settings also. If any one of you is interested, there are a lot of courses online. You can start learning those things. You can start doing those things also. I just wanted to give you an example of it. Then there are a lot of studies apart from our studies that we have done, that are done in all the fields that I had told you before. Epigenetics, there is study that methylation is one process which binds the DNA into the cell. When the DNA is coiled into chromosomes, the shape in which it is coiled, that shape is defined by methylation. There are methylation proteins which fix that DNA into shape, into different positions, and that those methylation proteins can be affected by different things. So this study has worked on that, experimented on that. Microbiome, I was telling you about. This study these people are studying microbiome, gut microbial diversity. What is the difference between Prakriti individuals in gut microbiome? How is their gut microbiome different? This is genome-wide correlation. The genome-wide means the whole genome was sequenced. This was done in genotype. Only the specific genotypes were taken and seen. This is the study that I told you about, first study that we did. This is the EGLN1 and this is the EGLN1 and VWF study. Apart from this, we also do study in response to stress. Now, what is response to stress? Response to environmental stress can be physiological level also and cellular level also. We can see that at physiological level also and cellular level also. Physiological level, I gave you an example of hypoxia. like oxygen levels. So when people will go there, they'll see that that is different thing. But we can create lab environments also where we can subject individuals. So there are hypoxia chambers where we put individuals or oxygen masks where we define the rate of oxygen, that here it is 21%, but as you move above sea level at 1000 meters above sea level, 2000 meters above sea level, the oxygen percentage decreases by 2%. So that you can decrease the oxygen level in lab settings and see if that individual is responding differently to that oxygen decrease of in oxygen level. And if something bad is happening, you can stop the experiment and revive that individual. That is totally experimentally controlled. So these kind of experiments are done in hypoxia. Then there is orthostatic stress. Orthostatic stress means if somebody is sitting or lying down and they get up immediately, then that change in position will give some kind of stress to that individual. That stress can be different in different people. So what we do is we take a table, make that person lie down on that table, and that tilt it like this. That person will be strapped to the table, tied to the table with straps, and then we tilt the table. When we tilt the table, we see that how the functions of that body change. Heart rate variability, blood pressure, SpO2, all these things are monitored. Whatever machines are there in ICU, all those machines are used, and all the vitals, all the physiological parameters are measured. What kind of change is seen in some individual? So this study we have seen is cough individuals respond late to orthostatic stress, and what individuals respond very fast. This is what we have found in this study. Then there is cellular level. Cellular level, I already told you, we take cells from different types of people. We culture those cells in lab and see that what is the effect. So what we do is we give UV shock. UV is also a kind of stress. We give UV and see that due to that ultraviolet rays, how much is the cell death? How much is the recovery of those cells? Uh, how they are growing, how good they are growing when there is external stress like UV or heat shock. Heat shock means higher temperatures than there are in the human body. 37 degrees is the normal temperature for cells to survive in our body. If you give heat shock, you give it 40 degrees, 45 degrees, 50 degrees. So what happens to the cells at cellular level when you give this heat shock? Or at cellular level, if you give 0.2% oxygen, 1% oxygen, 2% oxygen, 3% oxygen, different types of oxygen levels, if you give in cells, what happens to them? How fast they die, how fast they recover, how fast they become healthy, all those things, and what are the cellular responses. So, in this case, also, what we do is we take those cells, whatever are treated, we crush those cells, take the RNA out, and check how is the difference in biological processes. What is the difference of all these what do you say gene expressions? What is the difference in gene expression of those cellular components? Like what processes are affected through hypoxia, what processes are affected through heat shock, what processes are affected through UV. These all things are seen, and what are the differences amongst prakriti individual specific cell lines. Then, what are the applications of ergonomics? Ergonomics can be used for personalized nutrition and lifestyle recommendations. What a person should eat, how a person should work out, how a person should sleep, how much should that person sleep. If that person, if day sleep is good for that person, night duties are better for that person, if that person can handle night duties or not. All these things can be done using ergonomics if we have all the data related to it. Then there is disease susceptibility and prediction. Just a second. Disease susceptibility means what is that person prone to? What diseases this person is likely to have, so that we can predict them first and then prevent them. First, prevent the disease. If in case that person gets the disease, we can prevent the progression of disease also. Like uh, if somebody has got diabetes, then that person can be catered to a specific personalized nutrition, lifestyle, drug, everything, so that that disease does not progress. That disease is captured at where it is at. Then there is precision intervention, drug doses and selection, and there is promotive and positive health. Challenges, there are a lot of challenges when we integrate modern medicine into Ayurveda, because there are no previously standardized protocols. Every new experiment we plan, we have to standardize definitions, we have to standardize the protocols, we have to standardize everything. Then large scale studies are need to be done, because Ayurveda, what happens is at small basis, small studies are done. Those studies don't give enough evidence, enough data, so that we can bring out conclusions. All those studies that I told you about were done large scale studies. They were studies that included cohorts all over India, 5, 6 cohorts that had individuals, 2,000 to 10,000 participation individuals that were recruited and that were tested. So we need this amount of large data. It can be costly. It can be time consuming. So we, as individuals, alone we can't do anything. We have to be associated with organizations that do these things, and we have to be associated with uh, government organizations, with big research organizations. We need to apply to those places and go and work for them, so that we can do these studies, because individually we can't do these big studies. Each one genomic, one kit for one sample also costs lakhs of rupees. Then there are ethical concerns for everything that we do. Even that orthostatic stress, where we are not doing anything, we are just tilting it to 60 degrees, the table. That individual is lying down, no invasive procedures are done, only some ECG electrodes and BP machine, and these things are put on in that individual, and we are just tilting the table for 60 degrees, and we are asking one Prakriti question. This is what we are doing. There is no harm to that individual in all the tests we are doing. No invasive procedure, not even a single prick of needle we are doing. But still, we need ethical clearance for that. We need extensive ethical clearance. We need to do everything according to ethics, and we need to get it approved from everywhere. So ethical concerns are there. These things I'm telling you because you are going to go work in many organizations, you are going to do these things. So these things are which are necessary to be kept in mind. These are the challenges with that we need to overcome. Future directions, there are a lot of things that can be done. There are a lot of big institutions, NIH, NHS, National Institute of Health in USA, National Health Sciences in UK, Germany, all over Europe, China, Japan. All these countries are working towards personalized medicine. There are big industries, big geneticists, Ayurveda practitioners who want, they are data scientists who want data to work on. If you do these studies, you collaborate with data scientists, they'll they, they'll work on data and generate new things. Bioinformatics is a totally new branch that you can explore. You just need to learn some basic coding skills, and as you start doing it, you will learn more things. There are a lot of extensive things to be done in data, large scale data. If there are available large scale data, you, because every study that is done and published, they take all their data and put it into data repositories online. You can find data online, you can take that data and you can, this is what you can work on. If you are interested on your individual level, if you are interested in what do you think? What do you say? Bioinformatics or technological studies? Then this is what you can do. You can take courses online. There are a lot of free courses available. You can start learning, and then you can take data from databases, and you can start studying that data. You'll know how to do it. Once you start taking courses, you'll know how to do it. Then you can, even if the studies are done and data is there, you can find new things from that data. There is always possibility of finding new things. We have found one or two genes in that, you can find four or five more, and then you can give that findings to somebody else that can validate those findings. Some lab which has funds, they can do experiments and validate those findings also. So data, in field of data also, there is a lot of things that can be done. And in healthcare, if you are into healthcare settings, you can take all these researches, you can find online, you can search online, you can take the knowledge from these researches, and you can build healthcare models in your private settings also. You can just take a questionnaire, prakriti questionnaire. If you are just doing clinical work, you sit in a clinic, see patients, but before seeing patient, you make it a routine that you will do prakriti of every individual, and give directions to that individuals according to prakriti, based on whatever researches are done, based on whatever data is available online, whatever studies that you are seeing or reading. According to that, you can apply all these researches into your private practice also. If you are not doing any research, if you are in clinical practice also, then also you can update yourself to all the researches that are going on, and you can start implementing those findings in your private practice. That is what can be done. That is all from my side. Thank you. I will stop screen sharing, and if you have any questions, we can start that.
Okay, thank you very much for your nice presentation, and you are just elaborating everything in a very simple way. I think everybody has understand in each and every aspects, even the including the terminology of genomic medicine. So we will quickly take the questions. Anyone is interested, and before that, I want to explain that because this is like a very good model is advanced research in Ayurveda. So anybody can take this one because we are assessing the prakriti in our clinic, or we are assessing the prakriti in our OPD. So from that is a very preliminary level, and later on we can do advanced research. Like you can make a career of doing PhD in same institute, or you can make a career of a research organization. If you go to the abroad also, you can join to NHS and NIS. You can take this title of study and do the research. And so I think this is the good opportunity if you are planning to go abroad and doing the research. And another thing is these types of research can be easily published in good impact factor, because we never things like our article will be published in Biomed Center or some Lancet or other things, but such types of study will be easily published. No, it's not easy, but it can be unstable, and it can be published in Lancet or high impact citation journals. So this is the good opportunities, and as explained by the speaker, you need some of the good organization to work, and you need to work a lot. I think because Deepsika is working there for 4 or 5 years continuously, and at the end, you are able to publish like only a 4 or 5 paper, right?
Yeah.
Yeah. So, but the impact is very good. We never imagined to publish our articles from Ayurveda or like from our side from Nepal in those journals. So, we will quickly take one questions from our participant. You must ask some questions because she's explaining everything in very easy way, and trying to make you understandable in every aspect. Anyone? I think Jyoti has some question because she's interested. Okay. You can unmute. So anybody has planning like, okay, you are interested on this topic? Because
You're not audible. I think it's mute or something.
Jyoti is mute. I think she has some problem on her mic.
If your not microphone is not working, you can write in the chat also.
Yeah. Okay, so Susma Casey is also there. I think if she has not problem with her mic, she can raise a question. And you can raise your question in Nepali, no worry for that. Ashish is there.
Am I audible?
Yes, yes, you are audible.
Ma'am, you told about the features specific to Prakriti, like some fibrinogen synthesis like that. Is
Yeah.
it possible to make medicine also based on Prakriti or not?
Yes, it
For
is
me,
possible.
can, but specific Prakriti-wise medicine, is it feasible or not?
See, right now researches are very less. We have only few things which we know that okay, this prakriti has this thing. This prakriti is affected by this medicine. When we have a large data set, when we know all the Ayurvedic, there are a lot of herbs. Okay, all those herbs have chemical constituents, but those chemical constituents are not mapped to any kind of biological processes right now. If they are all mapped to biological processes also, it is a long shot, but from start, slowly we can go forward to it. Like in our lab, this study was done. First what we did was, um, there is pith of differences in high altitude adaptation, EGLN1 gene, I told you, right? Then what happened? Adatoda vasica, you know, vasa,
Yes.
adusa vasa, there is this drug, herb. This is a pith cough modulating drug. It works on pith cough. So what we did is we used the extract of that drug, and we did mice studies in which the mice were given, they were induced asthma, and it was, this steroid resistant asthma, like the asthma in which steroid also doesn't work. In that asthma, uh, pith cough is the factor that is working in hypoxia, and we took a pith cough modulating drug from Ayurveda and we used in that mice model of asthma, and we saw that not only it reduces the symptoms of asthma, but it also, what it does is it improves the mitochondrial framework also. What happens in asthma is in the cells, the mitochondria and the mitochondrial DNA also gets damaged. We have seen that Adatoda vasica has improved even the mitochondrial damage. Then what we did was we used those in cell lines. We started using those in cell lines, and when COVID came, in COVID, what was happening? Uh, Pneumonia. Due to pneumonia, there was breathlessness, low oxygen levels. So COVID was also, um, specified by hypoxia like conditions. So in hypoxia like conditions in COVID, in moderate to severe cases, in All India Institute of Ayurveda here, we started giving Adatoda Vasica to COVID individuals, the people who were admitted there for COVID, and we saw that this started working in COVID also. So we started from gene and we came to disease just by linking pith and cough to hypoxia. This is one example. Similarly, you can, if more studies are done into various other diseases or other signatures of Vat Pitta Cough. So what we are doing is we are mapping Vat Pitta Cough signatures to whatever is going on in our body, biological processes. Then we are taking those Vat Pitta Cough modulating drugs and using in conditions which are already known to have those biological processes disturbed. For example, hypoxia here, and we can finally use it in a clinical setting to get desired results.
Okay, that's I
Thank
think
you.
that's a good reply. And yeah, you have most more? Yeah, Susma.
No, sir, this is.
Okay, I have posted one link because usually what we can do in Nepal, so there was like one study and it was like on the Sherpas people, right? So
Yeah.
in where the higher altitude person, I think because there are a lot of study going on, and for Newari people, for ethnic people, there is like in 1985 and 1990 there was some study done, but recently after 2000 it was not being conducted. But Sherpas people, it has been continuously being done. We can see the article of 2008, 2017, and 2020. I think so many, a lot of researchers are interested on it. So any, if we can make some concept on it and we can go for some of the researchers in collaboration, I think this study can be done in Nepal also. So anybody is interested, you can do doing
Yeah.
on high altitude surpass.
So the example of the study that Prerak you have posted here, it is a purely genomic study. What they are doing is they are associating insertion or deletion polymorphisms in individuals, in Sherpa individuals particularly, to the angiotensin converting enzyme gene, like how the high altitude adaptation affects the angiotensin converting enzyme through the genetic polymorphisms. Now, what can be done is, if amongst Sherpas also, are one kind of people, but in that also there will different prakritis. If you see prakriti wise, so amongst them also, is there any difference? This can be seen, or or maybe something else, like in their um, if some disease connection is seen in that, then those cell lines can be taken and those cell lines can be looked at in case of some ayurvedic drug that works on angiotensin converting enzyme and how it works because of that insertion deletion. So all these studies can be framed around this study also, since these kind of studies are going on there in Nepal.
Okay, I think you know everyone is known to Dr. Basanta Panta, and he was also planning some experimental research, like because he has a very good cancer cell institute, Annapurna Cancer Neurological Cancer Institute, and he also planning on the same way. He wants to like, he has a lot of cases of those patient, and he wants to take those cell line, and he wants to expand with Ayurvedic products. I think also we can also do such expect. And okay, so I want to take with Leon, Leon Porter. I think he's so much curious about this thing. And do you have anything in your mind? Like you can share your what was the session going on and what's in your mind? Like you are thinking something about it?
उम्, मैले त म्यामले पहिला एक्सप्लेन गर्नुभएको थियो त्यो प्रकृतिको चाहिँ त्यो मेसिनबाट कसरी पत्ता लगाउने त्यो चाहिँ प्र्याक्टिस भइरहेछ कि त्योमा गरिरहेको छ कि अहिले के भइरहेको छ त्यो कुरा चाहिँ
uh,
Deepsika, do
huh,
you understand it?
I understood that prakriti,
त्यो
if prakriti can be like, if prakriti is given anywhere, or what,
अ,
can you
त्यो
repeat?
Yeah, he was
ल-
trying. He was trying to say that the Prakriti you have mentioned, the
Yeah.
experimentation process, the machine learning, and all the laboratory process, genomic laboratory part, it is going on.
Yeah, it is going on, still going on. A lot of studies are going on, Abhi.
So can you just like elaborate, like some elaborate over this thing? I think he's not on. He's not clear about it. That's his question.
Okay, how it is done is what he's trying to ask, right? So, what is done is, we take individuals from different, we take healthy individuals who have no known disease, and we take those individuals and we ask them a set of questions that determine their prakriti. And if we see that some individuals have a higher proportion of one type of uh, dosha in their prakriti, like vata dominant, pitta dominant, or kapha dominant, those individuals we take blood samples from them. We do different kinds of tests. We take blood samples and do biochemical tests, hematological tests, and genomic tests. And the results in those tests, we see that if there are any differences between the vat group, pitta group, and kapha group, and how are they different from people who have mixed kind of proportions of prakriti.
Yeah, I have visited her institute, and there are a lot of sophisticated laboratory for analyzing the genomic level, because you may be interested on PCR, okay, polymeric chain reaction machine are there, ELISA are there. I think he is trying to put those question. So there are a lot of instrumentation available there, and you can minutely do a different level of studies, even not only genomic level, you can go for like epigenomic level also. So in very minute level, the instrument are available. Yeah.
It is a very big laboratory. So our institute is kind of the biggest and the best biological institute in our country. So we have all kinds of instruments from basic biology to high throughput gene sequencing. We have sequencers where we can sequence the genes, we can sequence the RNA. Then we have basic instruments, all of them are there. Cell culture instruments, when we need to incubate cells, we need to propagate cells, culture those cells. All the cell culture facilities available. Even all kinds of sequencing, microbiome sequencing, human genome sequencing, sequencers are there. Portable sequence, big sequencers are there. Small portable sequencers are there. There are genotyping platforms also. Like different companies have their own set of genotyping platforms. They have specified chips, genotyping chips that we get, and their machines are there. So all those things are available in this institute.
Okay. So I think we have already crossed our timeline, and thank you very much for your session. And so if anybody getting the research idea on this topic, she will be providing the mentorship for you people. So you have to come back with some of the research idea, small level studies, and so that more and more you can do in your future, and in collaboration with some of the organization, or you can move to some abroad and for this study with those proposals. Okay, thank you for your time, and thank you all the participants for joining and listening and putting into the discussion. Thank you.
Thank you. Thank you, Prera.
Thank you, ma'am.